All right, for the last two. So for computational methods, our goal is to try to build models that'll take us from genotype to phenotype that are interpretable. We talked some about that we don't want these black box technologies anymore, and we may want them to be either mechanistic or causal and predictive. And so some of the things we talked about that are important resources that we need to be moving toward are structured models that allow us to move from the gene level to higher order levels. This came about in the first day when Emma Farley presented this, going from reading kind of letters. If you take that concept and think about reading, the letters are variants, and we want to be able to read words, which are genes, sentences or functional units, paragraphs or networks, and images are cell biology. We have a lot of existing data that we primarily look at in the terms of the variants and perhaps the genes or the gene regions. We need to get to where we're doing the analyses in terms of the functional units and the networks. And so even with existing data, we need methodology to try to look at those combinations of variants and how they work together to lead to phenotype. We need systematic phenotype extraction and mapping from electronic health records, and as was talked about yesterday, we need to do that in collaboration with clinical informatics experts. There's a whole field of people working in this space, so we don't need to reinvent the wheel, but we need to engage with those experts who don't have the genetics and genomics expertise, and yet those institutions that have that expertise are building biobanks, and so partnerships with those communities is important. We need structured featurized polygenic risk score models, as well as evaluating these PRS in diverse populations. Most of the PRS have been generated in individuals of European descent, and so how do those PRS transfer to other ancestry populations, other kind of slightly different disease symptom populations? And we want to go beyond just adding SNPs together, but thinking about including environmental factors, clinical factors, multi-omics factors in those polygenic risk score models. We need methods to compute on private or encrypted data. This came up a few times, that there's certainly in imaging data, it's very difficult to de-identify. In medical images, individuals' names are on the PDF of the image, and so it's very hard to scrub those in order to make them de-identified. But also in terms of data sharing and privacy, if we want to be able to upload data into these cloud-based platforms and compute on them, we may need to be able to develop ways to do that in an encrypted way. We need model-driven experimental design on all of these perturbation experiments that we talked about, especially the interactions or combinations of perturbations, so that we can maximize inference, and effective methods to analyze and interpret structural variation. And we should have added, and those hard to sequence regions, once we get the technology to sequence them, we're also going to need the computational tools to align them and call variants and interpret them. And then last, the data platforms. We need scaled, accessible data platforms. So we're going to generate lots of data and lots of tools, but if they're not accessible to the community, then we've done a huge disservice. So we need both the genotypes and phenotypes to be accessible. We think it's really important that we think about interacting data platforms. The kiss of death of any data platform is that you try to build one platform that does everything. And so if we focus the platforms but make them interoperable and make that be something that we start with in the initial planning and make those platform communities talk to one another so that we don't end up with 2 or 3 platforms that can't communicate. If we do that from the start for both the genome processing and the analysis, that's going to be really important. And that includes the genotype information, the molecular phenotypes, imaging, the associations that we're finding, wearables, environmental exposures, electronic health records. This idea of bringing the algorithms to the data, but also making where those platforms are, be able to communicate with one another. We need a data portal with genetic variants and associated evidence from comprehensive PheWAS. This was actually something that was in the Excel sheet from the surveys that were done before the meeting, but this idea that a lot of PheWAS studies have been done, and if we could get all of those into one database, that information would be useful even for the bench scientists who are doing CRISPR experiments. So imagine that you've done these really interesting cellular model experiments, and you have a list of variants that you found are functionally interesting in a cell. And now you wonder, do they mean anything for human health? Well, you could take those variants, and presumably a lot of them have already had a PheWAS in hundreds or thousands of people, and you could see, in order to prioritize which of those variants do I move forward with, which ones are related to phenotypes, which ones are expressed in a tissue that would make sense given the phenotype that you're seeing. And so if we had all of that information aggregated, it would give us a lot of power. A visualization platform, this came up a bit yesterday, but we think this is really important for this multimodal data, and tools to explore and visualize the data. This was the Alexa, show me this region. Alexa, show me what's interesting here. If we can engage data visualization scientists, artificial intelligence experts, and the genome scientists together, and I think we heard a workshop mentioned. I think that would be a great start to get people in the room, but getting them to work together. Because if genome scientists try to create data visualizations, I mean, there's a whole field that has already done that. We should bring that field to us, but they can't do it without us because they don't understand the data and how to make sense of it. So that we think is a really important collaboration. And even more important than, if we generate a lot more data, we're going to be in trouble because we can't even look at the data that we have now. So that's something that we should prioritize even sooner than waiting for all of the data. We should start with the data we have. Platforms for the direct-to-patient work, including, this came up once or twice in different sessions, this idea that there's a lot of data being generated outside of NIH and outside of research, and it's going to be very hard to get the commercial sector to share their data with us. But the patients often get their data, or the research participants. If they have a way to bring the data to some resource. We heard yesterday, research participants would be appalled to know how little their data are being used. They think their data are being used broadly. So if we build a platform that they can upload or send their data and then contribute whatever phenotype information we would want to collect, that would be a way that we could get access to some of that data that's being generated out in the world. And then the last one is a unified public repository for the predictive models of genomics. So this is something that Anshul mentioned yesterday, similar to how we have PubMed for publications or GEO or SRA for data repositories, some way to link all of these different data repositories and publications and being able to kind of cross-cut across these different repositories. And that's it. So I think we can open up for discussion.